Hi, SUPA people. All right. Welcome to another edition of SUPA Chemistry. Today we are going to finish organic chemistry. And actually, we're going to deal with the bridge between biology and chemistry. All right, so we're going to deal with what's called biochemistry. And when you're in college, biochemistry is actually a 4-semester course that you can actually take. If you're a bio major, you'll need to take this. If you're a chem major, you'll probably have to take this too. We'll just touch on some of these organic molecules that have to do with living organisms. And a lot of if you took AP Bio, a lot of these you'll recognize. All right, so first of all, the ultimate source of energy for here on Earth is our sun. Okay, so the sun really provides all the energy that living organisms here on Earth use. Now, there are a couple exceptions, like some of the deep sea vents, that heat that's provided by the Earth, all right, maybe provides some energy, but ultimately the sun is really what gives us all our energy. So that sunlight that hits the Earth has to be harvested, and it's harvested through photosynthesis. So photosynthesis provides the energy for both plants and animals. So let's talk a little bit about what's called biopolymers. In last class, we talked a little bit about polymers, and these are defined as biological important polymers. And these include, or at least ones that we're going to talk about, proteins, and it's EINS, polysaccharides, which are carbohydrates, and finally our nucleic acids. So these are our different biological polymers that we're going to discuss. So we're going to go through proteins first. So let's label this A. So our proteins are the structural component of all tissues. Okay, so in both plants and animals, our proteins make up all the structural parts. So all of our organs, skins, hair, heart, leaves, okay, they're all made up of proteins. Proteins also catalyze a lot of our biological reactions. So a lot of our organic reactions, the ones that we talked about last class, they're actually pretty slow. And if they're not catalyzed, we wouldn't be able to use them in our body. And proteins will help us catalyze a lot of those reactions. Another use of proteins is oxygen transport. Okay, so the proteins, hemoglobin, and that kind of stuff right? They help us transport oxygen. Hormones are proteins. And, oops, this is a 5. What makes up our proteins? Okay, what comes together to form proteins? And these are our amino acids. So these are composed of amino acids. Now, okay, that should look familiar. We know what an amine group is. We know what an acid group is. Right, so an amino acid are building blocks of protein. So let's talk about those amino acids a little bit. So let's talk about alpha amino acids. And these are the building blocks of proteins. So the amino acid tells us that the molecule has a carboxyl group. That's our Cu group. And an amine group. The alpha part tells us the amino group is on the carbon, on the carbon backbone. And it's immediately adjacent to the carbon with the carboxylic acid group. So let me give you an example of an amino acid. And this is one of our primary ones. This is alanine. So alanine is actually a 3-carbon chain, and it's got a carboxyl group, and it's got an amine group on the carbon adjacent to the carbon with the carboxyl group. So it would be on this carbon. And then we need our hydrogens. Right? So this is an example of an amino acid. Here's my carboxylic acid group. This is my carboxyl group. And this is my amine. Now, it's an alpha amino acid, okay? And that alpha is dictating where this amine group is. Right? So if this amine group was over here on this carbon, all right, it would not be an alpha amino acid. So now we link these amino acids together into long chains, and they're complicated. And those long chains form our proteins. So our amino acids are linked together into proteins by bonds that are called amide linkages. And these amide linkages also have a special name called peptide bonds. So let's go ahead and look at this peptide bond and how we form this polymer, or this biopolymer. So the peptide bond, or the amide linkage, is formed between the carboxyl group of one amino acid and the amino group of a different amino acid by condensation polymerization. And we talked about condensation polymerization. So remember, polymerization is making those long carbon chains, right? It's making a polymer. In condensation polymerization, it's also making water, right? So let's look at glycine and alanine. Okay, so glycine is this. It's got our carboxyl group. It's got the amine here, right? And this has two hydrogens on it, right? So this is our glycine, right? And let's link it with an alanine, which would be something like this. We got our three carbons, oxygen, OH. Our amine group is here. And we got three hydrogens and a hydrogen. So what happens during the condensation polymerization, when we're making our protein, the OH from the acid comes off. One of the H's comes off, the nitrogen, and these come together to form our H2O. And then these are linked here. So I'm going to rewrite this. NH2, and then we got CH, then we have C, double bond O. This comes off, and then this goes on. So this is off, so this is linked to a nitrogen. And then we have a hydrogen coming off of that nitrogen. It's connected to this second carbon right here. And then what happens is off of that carbon, we also have a CH3 group. And then we have a C double bond O, OH. And this is what it would look like in the protein when these are linked. Now, where's my peptide bond? Well, my peptide bond is right here, where we link them. So this is my peptide bond. But it's also an amide linkage. It's an amide. Okay, so if you remember what an amide was, we have a double bonded oxygen on a carbon with a nitrogen coming off of that. Okay, so this is actually an amide, and we call it an amide linkage or a peptide bond. Now, what would happen is we would have another one here. It might, another amino acid Okay, and it would do the same sort of thing. The OH would come off, it would link to the nitrogen, all right, and we would get another one. And this grows and grows and grows, and this is how we make our proteins. So what this turns into is something called a polypeptide. And this is formed when a large number of amino acids are linked by peptide bonds. Okay, so we link a lot of these, we get a polypeptide, and a polypeptide is an example of a polypeptide would be like a protein. Right. So now proteins, or these polypeptides, they have structure. First of all, they have primary structure, and the primary structure is the sequence of the amino acids, the actual order of amino acids. And it's the sequence from the N terminus which is the N terminus is the amino end, to the C terminus, which is the carboxylic end. So in our short polypeptide that we made, this would be the N terminus, okay, where the amine is, and then this would be the C terminus. So our primary structure is in how these are put together, what exactly amino acids we have, and it goes from the N terminus to the C terminus. Now, changing one amino acid can alter the biochemistry of the protein. If we just change one of these, it can change the whole protein and make it not effective, right? It may not be able to carry oxygen in our blood, all right? It might not be able to catalyze a reaction, all right? So just changing one of these in the primary structure, it can change the whole protein. So proteins have primary structure, and then they have secondary structure. So once we start getting this sequence of amino acids, the protein chain orients themselves into regular patterns. And the proteins are going to orient themselves into alpha helixes into an alpha helix or a beta sheet. Okay, so these alpha helixes, so basically what happens is our amino acid groups it's gonna fold up on itself, and we're either gonna fold it up into an alpha helix or a beta sheet. Right now, as it folds up, the amino acids are gonna interact with each other, and the secondary structure is held together with intermolecular forces. And for the most part, they're either hydrogen bonds or they're dispersion forces. Okay. So the first kind of fold, the first organization, okay, once we have the primary structure, that first folding, it goes into what's called an alpha sheet or a beta sheet. And then these alpha helixi or beta sheets actually fold together again, and they form our tertiary structure. So the alpha helix and the beta sheet fold up into a specific three-dimensional structure. And this is mostly based on electrostatic and van der Waals forces. Okay, so we get this second folding. And I have a picture of this. Where did I put it? Maybe I have it. Let me see if I can find it real quick. So this is out of the textbook. All right, so let's just look real quick. I had a better picture of this, but since it's here. All right, so here's my primary structure. So here are my amino acids. Right, so the primary structure is those amino acids and how they're put together. And then those amino acids, okay, once they're all together, this primary structure, it forms either an alpha helix, and that's what this is, right? Or it'll form beta sheets. So this is an example of an alpha helix, right? So it starts to fold up on itself and we get this alpha helix. That's my secondary structure. Then this secondary structure folds up again. So once this is long enough, it folds up again and we get tertiary structure. Now, if we have enough of the protein, multiple proteins will come together. Okay, so we got multiple proteins come together, and they actually form what's called quaternary structure. And it's got to do with all of our foldings. So it's a pretty good example. Right? I had another one. I don't know where I did, but I did with it. Right? So when we're talking about these structures, we're just talking about the different foldings, all right, of each one of each part of the protein itself. All right, so let's talk about carbohydrates next. So carbohydrates actually tend to have hydroxyl groups, aldehydes, and ketones. And okay, so our carbohydrates are what's storing a bunch of our energy, all right? So we use a lot of this for energy storage. Do you remember what's the most famous carbohydrate? Glucose. Right? So glucose is the C6H12O6. So our formula for glucose is a ring, but there's an oxygen in the ring. So we got C, let's see, C, O, C, 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 C. Here's my ring. And then off of this carbon, we have a C, and this has an OH group on it. And then this carbon has an OH group off of it. And then this has an OH, and we'll put an H on there just to keep it out of the way. And this has an OH here. Technically, we really wouldn't put the H's on the inside, but I want you to see these OH groups. All right, this is going to have an OH. We'll put the H over here so it's out of the way. Okay, so this is glucose. Okay, and there's something that's very similar to this, okay, that's also a carbohydrate. And it's similar, but it looks something like this. So instead of having 1, 2, 3, 4, 5 carbons in the ring, this one only has 1, 2, 3, 4 carbons in the ring. All right, and it differs this way. So this is going to have, this still has a C with an OH group this way. This still has my hydroxyl group, and this still has a hydroxyl group with an H. But this carbon has another carbon coming off of it with an OH group here. Okay, the oxygen is set. Okay, so really kind of what we've done, all right, is instead of this oxygen going to this carbon is kind of kicked out and it goes to this one, right? If you can kind of see what that does. Okay. This carbohydrate is what's called fructose. And these together, hopefully you remember, are called monosaccharides. ARIDES. Monosaccharides. Mono means one, right? And these are our simple sugars. Now, we can take these simple sugars and we can polymerize them. We can link them together. Okay? And if we take two monosaccharides and put them together, they're going to form what's called a disaccharide. So disaccharide is when we take two monosaccharides and we link them together by dehydration synthesis. So examples of disaccharides. Okay, examples. The two biggest ones are sucrose and lactose. So these have two monosaccharides linked together. And sucrose, for those of you who don't know, is just table sugar. And lactose is what's in milk, the sugar in milk. Okay, so these are disaccharides. Remember, this is okay. We can break these down, and it's where we get energy from. Now, if we link disaccharides together, or just multiple monosaccharides, we can form polysaccharides. And a polysaccharide is really just a polymer of monosaccharides. So we take those glucoses and fructoses and we link them together, right, into long carbon chains. And this is where we have long-term storage of energy. All right, so examples of polysaccharides, okay, starch, right? Glycogen and cellulose. Okay, so these are just examples of polysaccharides. And all we're doing to make these polysaccharides is we're just taking these, where did I put this? We're just taking these monosaccharides, okay, and we're going to link them through dehydration synthesis. So we're going to pull an OH group off. We're going to pull an H group off. I'll pull an OH and an H. Water will be given off, okay, and they will link. Okay, they'll link. Right? And then if we have lots of them, okay, we'll form that polysaccharide. And we can just store tons and tons and tons of energy in this. All right, so those are our carbohydrates. Our next biomolecule that we're going to talk about are lipids. And our lipids are nonpolar biological molecules used for long-term energy storage. Okay, so our lipids are nonpolar biological molecules for long-term energy storage. Now, I want to switch this over so we have a bigger sheet here. And the first lipid I want to talk about are our fats. So in fats, we take glycerol. And it reacts with a condensation reaction with a fatty acid. Now, let's talk about glycerol for a second. Another name for glycerol is 1,2,3 propentriol. 1,2,3 propentriol. So glycerol, and we made this in the lab. Okay, so it's a 3-carbon chain, and it's got an OH group coming off of each one. Okay, so this is our glycerol molecule, right? So we're going to take this glycerol molecule and we're going to react it with a fatty acid. So what we'll do is we're going to take this and we're going to react it with a fatty acid. And a fatty acid, Okay, matter of fact, we need 3 of them because we're going to put one here, we're going to put one here, and we're going to put one here. So we need 3, and this is the molecular formula of a fatty acid. It's C15H29 COOH. So it's got an acid group on it. It's a fatty acid. So this is my fatty acid. Okay. So these are going to link with a condensation reaction. So what we end up getting is we get our glycerol. The oxygen stays on, right? The H comes off, and then what we get is this long carbon chain, 2, 3, and I'm gonna put a dot because this is 15 carbons long, and then on the end we get C double bond O OH, and this is the same thing. Dot dot dot. C double bond O OH, and this is the same thing. C. So you get these long tails, right? So this right here is a fat. Now remember, okay, well, not remember, but there are lots of types of fats, okay? And some of them are good for us and some of them are not so good for us, right? So I'm sure you've heard of saturated fats. Well, you know what saturated means. If a carbon atom is saturated, okay, it's full of hydrogens, it's got no double or triple bonds. Okay, so if it's a saturated fat, that tells us that it has no double or triple bonds in the carbon chains. They're saturated fats. What if we have unsaturated fats? Well, if they're unsaturated fats, they're going to have double bonds. Not so many triples. And because they have double bonds, an unsaturated fat actually has another name. These are also called cis-trans fats. Okay, because we get those cis-trans isomers. Now, we can take it one step further. You've probably heard of monounsaturated fats. Okay, well, what does mono tell me? One double. Okay, so these are carbon chains that have one double bond in here. Okay, so there's one double bond in this carbon chain somewhere. Or we'll have polyunsaturated. Where we have many double bonds. So I'm sure these are all things that you've heard of, and it's really got to do with these carbon chains. All right, so the other lipid that we have to talk about is really similar to fats. And these are called phospholipids. And our phospholipids are what forms our membranes. So these are very similar to fats. we're going to still start with glycerol. We need 2 fatty acids this time, and 1 phosphate or polar group. So what we end up with is something like this. So here's my glycerol, and we have O's here. But in my phospholipid, the O comes over here, and we get this phosphate group, and it looks like this: P, O, O, double bond O. And then over here, we get a long carbon chain. Okay, we get a fatty acid. And in our membranes, we want one side of the membrane to be hydrophilic and one side to be hydrophobic. Okay, so one side needs to be water-loving, the other side needs to be water-hating, right, to make that membrane. This side of the phospholipid is our hydrophilic because of all these oxygen atoms, okay, and it can form hydrogen bonds and stuff like that. This guy, where we have all these carb chains, is our hydrophobic. Okay. It's non-polar. It doesn't like that polar water molecule, so it pushes. They repel each other. Right. So this hydrophilic side, okay, loves the water. It's attracted to that water. Lots of hydrogen bonds. Then this non-polar side of the molecule is hydrophobic. All right. And it pushes that water away, and we can form membranes from this phospholipid. All right, so if these join together, okay, they join together, we get one side that's hydrophilic, and we get one side that's hydrophobic. All right, so the last thing we have to talk about are our nucleic acids. Okay, so our nucleic acids are biopolymers that are chemical carriers of genetic information. So this is our DNA and our RNA. So if we have a polymer, that means we have monomers. Okay, so monomers of nucleic acids are called nucleotides. And these monomers consist of a 5-carbon sugar, a nitrogen base, and a phosphate group. So what it would look like is something like this. We got a 5-carbon sugar with a nitrogen base and a phosphate group. So our 5-carbon sugar would look something like this. And then coming off here, we'd have a nitrogen base. And then coming off of here, we have an O, P, O, double bond O, and an O. And this right here is my phosphate group. Now, that would be linked to another one. And this would have a nitrogen base. And the O would be coming off of here with a P, double bond O, O, O. And again, that would be linked to another nucleic acid. Okay, this would be my nitrogen base. And this would be O, and this would keep going. Now, how do these come together? Well, it comes together with a condensation reaction. Okay. So this right here would be part of a polynucleotide. Okay, this is be part of your DNA or an RNA. Okay, so this would be part of a polynucleotide. All right, now let's talk about this nitrogen base because this is important. All right, so this is our adenine, guanine, cytosine, and thymine in our DNA. Guanine, cytosine, and our thymine. All right, so remember, if you remember back a bit, bio, we got A's and G's and C's and T's, and these match up with each other, right? So this is, okay, the G and the C pair, and the A's and the T's pair. And what happens, okay, so we have one nitrogen base on this side, and then we would have a nitrogen base on this side. And it would have this 5 carbon, we have the same thing on the other side. And this would form that double helix. Okay, so you get this interaction here. Now, these are in DNA. There's another nitrogen base in our RNA, and that's the uracil. Or a U. This is how our DNA or RNA comes together. Right? So we have this 5 carbon sugar, we have that nitrogen base, and it's either an adenine, guanine, cytosine, or thymine. All right, and then it's linked together with these phosphate groups. Right, so let's see if we can draw what this might look like. So we would have O, oops, and let's say this is an A for our group, right? And then this comes down here, we have an O, P, double bond, O, 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 and then this is linked again. And let's say that this is a G, and then this comes down with an O, P, double bond, O, 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 and this would be linked maybe we have I don't know, maybe a T. Okay. Then on the other side, okay, and that would keep going. This would have an O down here. Remember, the A goes with a T, so we would have an intermolecular force, and we'd have a T, right? And then this is attached to the would have an O, P, double bond, double bond, O, O, and then this would attach to, it's kind of hard. I should have put this over here. And coming off of this guy, I screwed it up. That's okay. Okay, this would have been a C sitting here, and those would have linked. Okay, and this would have an O coming off of it, right? And that would have a P double bond O, right? And then this would link again with another one of these guys. And coming off here would be an A, and that would link with this guy. It doesn't show up very well. I drew it horribly. All right, but in the textbook it shows you this, and this is this double helix. That's bad. Sorry. All right, so that's here. We would have the AT combination. Here we'd have the GC combination, and then we would have the TA combination. And these nitrogen bases, or this order, right, is what ends up giving us, all right, the genetic information. And okay, and here we get this double helix. All right. Really, that's as deep as I'm going to go into biochemistry. You should know those things that we talked about, right? So make sure that you go through the nucleic acids, make sure you go through your proteins and your polysaccharides, all right? And make sure that you know those things, but that's really as deep as we're going to go. All right, so have a good afternoon, and we will talk to you soon.